Alright, so this video is meant to come after episode 1, so note that the clear label, this is Unit 7, episode 2. Excuse me. You should watch episode 1 in class. It's a little longer, it's about half an hour on the cell cycle and mitosis. And episode 2 of this one is about DNA structure and DNA replication. And this is what your homework is. You may have time in class to get this done. So the learning targets, A, B, and C listed here, are that you can draw or build the DNA molecule and identify its building blocks. B, you can understand how, and understand how DNA replicates, and to be able to explain it. You might understand it, but I don't really know that you understand it until you can explain it to me. And C, you understand why DNA replicates, and again, be able to explain it as a demonstration of your understanding. So let's go ahead and get started and dive right into DNA. Alright, so we've got DNA, and in your vocab list, you should already have written down the DNA stands for deoxyribonucleic acid. And yes, you do need to commit this to memory. So we've got deoxyribonucleic acid. So the D from deoxyribo, N for nucleic, and A for acid. That's where we get our DNA from, or the letters for DNA. Now DNA is made up of, DNA is a polymer, and it's made out of several monomers, or building blocks. And in this case, that monomer is called a DNA nucleotide. And the nucleotide is made out of 3 separate parts. It's got a phosphate group, a sugar, and a nitrogen base. Now let's say that this sugar, so let's go ahead and label this. This is our phosphate group. This is the deoxyribose sugar, and this is the nitrogenous base. Now, again, what I said was that this whole thing is called the DNA nucleotide. And so this is the monomer that makes up our DNA polymer. And so we're going to draw several of these together. And let's make it a ladder model. Alright, so what I've done here is I added 2 other drawings to our DNA nucleotide drawing. And I'm just going to label these as describing what kind of model it is. So this one in red is called the ladder model of DNA. And this one in blue and green is called the helix model of DNA. Excuse me. And basically they're the same thing, but just shown in different ways. So I'm going to go ahead and just color in orange in this model, in the ladder model, all of the deoxyribose sugars, just so you can see this DNA nucleotide in this drawing. So these things are all the deoxyribose sugars in DNA. And notice that there are 2 strands of it on opposite sides. We've got phosphates here that we'll color in blue on this model. These are phosphates, and these phosphates connect one nucleotide to the next in a line. notice this phosphate's connecting, this phosphate here is the same one here to this deoxyribose, and it's connected to the next deoxyribose. The same thing on the other side, but notice that the deoxyribose sugars are flipped the other way. The point on this side is up, the point on this side is down. And then all of these things in the middle, I guess I should use a different color. Let's use white. These are the nitrogenous bases. And there's something very, very important about them that we're going to talk about in just a few minutes. This helix model is more of the double helix. Because there are two sides to DNA, it actually wraps around itself because there are little hydrogen bonds, yes, kind of like in water, between water molecules, between these nitrogen bases that are connecting them. And they actually kind of torque or pull the model of DNA, or the structure of DNA, so that it twists. And so that's what the helix means, is that it twists. So these nitrogen bases are actually very interesting because that's the only thing that makes us different from all other living things. So as far as the nitrogen bases, there are only 4 different kinds. There are only 4 different kinds, which means that it's the order of these things that makes the big difference. And we've got, these are the 4: adenine, thymine, guanine, and cytosine. And what we know is that A and T bond together and C and G bond together. So if we go back, we know, and you might notice the trend here in these structures, that there's always a complementary base pair. If this was a T, sorry about that. If this was a T, this would be an A. If this is a G, this would be a C. Because this is the same shape as that C, this is a C, and this is a G. If this was, let's say, this is our A, this is a T, and this is our T, and an A. They'll always pair up like that across the structure of DNA, and that's really, really important for DNA replication, which is what you're getting ready for. So here's my question for you, and let's see if you can do it. If I gave you a sequence of DNA nucleotides on one side, let's say the order is A, T, C, C, A, G, could you, using those base pairing rules, tell me what goes on the other side? Now, just as a quick note, sometimes DNA molecules get abbreviated so that they kind of look like just this line with structures inside. You should know that this line actually, actually represents the sugar and the phosphates. Because those are the same in every single nucleotide of DNA, we're not so concerned about it. It's more of the nitrogen bases that are more important, so we draw those usually. So sometimes we draw DNA in a ladder kind of model, but instead of drawing every sugar and phosphate, we just draw it like this. And go ahead and try to fill that in, and we'll see if you're right a little later. Okay, so let's talk about how DNA replicates. And really what I want you to do before we do that is pause this video and look at Unit 7, Episode 1 podcast and determine and answer this question. When in the cell cycle does DNA get copied? And you might even be able to figure out why does it get copied. And just to direct you a little bit, look at mitosis. Okay, anyways, let's talk about DNA getting copied. So let's say that we've got our strand of DNA molecules. I'm going to write a sequence. And then the complementary base pairs. And there are hydrogen bonds between these nucleotides. And we've got our DNA strand. Remember, this line represents sugars and phosphates, but we just draw them as a line because they're all the same. It's these nitrogenous bases that are going to be key later in the process, later in our discussions through this unit. So what ends up happening is actually, if we understand that, the DNA strand will continue on. We've got millions, billions, trillions of nitrogenous bases. It'll continue on, and we've got more and more letters. But really what's interesting, and I'll just, what ends up happening is a chemical, an enzyme comes in and actually splits apart the DNA molecule into two separate sides. And so let's say if we continue this sequence, and then its complements were, and these were the complements before this molecule came in and split it apart, and now the hydrogen bond has been broken. And we've got these loose molecules, or unbonded molecules. And so what ends up happening is another molecule, another enzyme will come in and start adding free-floating nucleotides. So this T will get bonded to an A, this A to a T, C to a G, T to an A to a T, C to a G. And while it's doing that, for each one, it's adding the nucleotide. So not just the nucleotide, not just the nitrogen base, but the sugar and phosphate as well. And it does the same thing to this side. Now, if you look at these sequences that we wrote, if we look at the yellow letters, T to A to T, C to G, T to A to T, C to G, we actually see the exact same sequences here in the red. We've got T and A to T, C to G, T to A to T, C to G, T A T, C G, T A T, C G. Exact same combination. And so this process will continue along the entire strand of DNA until we get two copies of DNA. And if those enzymes, those molecules, did a good enough job of pairing up these nucleotides, they should be two exact copies. And that leads to the sister chromatids that you see in mitosis. And that's essentially the process of DNA replication. It's pretty easy as long as you understand what this model represents and the molecules that are involved in it. Alright, so let's revisit the loading targets. If you can draw or build a DNA molecule and identify its building blocks, make sure that you give yourself a check mark that you can do that. If you can understand how DNA replicates and explain it, give yourself a check mark. And if you figured out the all-important question of why DNA replicates and can explain it, remember this is actually going to require that you pull information from the previous video, the one that you watched in class, and connect to this video. So you got to make some connections to be able to answer this question. But if you can do it, give yourself a great big check mark. Remember to write down questions. Write down questions that you have, because that can lead our discussion in class. Okay? And that wraps it up.